So the last thing that we've seen together was the simple columnar epithelium, and we've seen how to identify this type of epithelium. Again, one row of tooled cells are lining this tube. So simple means one cell layer, columnar means I'm forming cells. Can you hear me? Can you hear me well? Can you hear me? All right, great. All right, so looking on here at another type of epithelium, you can see here, am I formed of one row of cells or more than one cell layer? What do you think? Do I have one cell layer or more than one cell layer in this tissue? Two cell layers? Yes. What do you think? One or more than one cell layer? So for the first impression I got looking at this tissue is that this tissue is formed of more than one cell layer. So I'm about to say it's a stratified epithelium. But this is not the case. So what we're looking at in here is just one cell layer, but because those cells are very close to one another, what you see on here are the nuclei of the cells, which are arranged on different levels. So when I look on here, I'll see this is one layer, this is another layer. But actually, this is just one cell layer, but the cells are closely attached to one another. They are very dense. So looking at this tissue, how would I be able to identify that this is one cell layer? I can actually see that all the cells are attached to the basement membrane. This is my basement membrane on here. All the cells are attached to the basement membrane, but I can't actually show you that each of them is going to be reaching the basement membrane. So how would I be able to identify this? If you remember the characteristics of the epithelium, epithelium didn't have blood vessels, if you remember. It was avascular. But how the cells did get their nutrients and oxygen? They got their oxygen and nutrients by diffusion from the underlying connective tissue. So what happens to a cell? If we look on here, that will be moving away from the source of nutrients and oxygen. Will it be large or small? What happens to you if you don't eat for weeks? You shrink, right? So the same concept for the cells. So cells, as they get less oxygen and nutrients, they will shrink. They will become much smaller in size. So we're looking here at this tissue. What do you think are those cells. A cell that looks like this. Is this a squamous cell or a columnar cell? What do you think? Is this a flat, cube-shaped, or tall cell? What this one? What's a squamous? Squamous means the cells are flat, so the width of the cell is much greater than the height of the cell. Does this apply to this cell? This is the height of the cell. This is the width of the cell. What do you think? Is this a flat cell? Is this a squamous cell? It's a columnar cell, you see? The height is much greater than the width of the cell, so it's a columnar cell. How about this one? How about this one? Squamous. This one is going to be flattened. The width of the cell is much greater than the height of the cell. So it's a squamous cell. Is there any logic in here to have squamous cells, small cells at the base, and large columnar cells at the apex of the tissue? Does this make sense? To have squamous cells at the base and columnar cells at the apex. Does this make sense? According to what we mentioned? What do you think? No. Why? Because if I have tall cells, if I have columnar cells, they were supposed to be getting much more nutrients and oxygen. So they should be at the base. They should be in direct contact with the basement membrane to be able to get nutrients and oxygen for them to be larger in size. Compared to a squamous cell is the one that's going to be located away from my basement membrane. So this will be of much smaller size. So looking at flat cells at the base and tall cells at the apex doesn't make sense. It doesn't make sense. So this simply will tell you that actually all the cells are attached to the basement membrane, but they are just of different heights. They are of different heights. So this is a squeezed cell. That's why it didn't look columnar. It was squeezed by the surrounding cells. So how would I identify this type of tissue? For the first impression, I will identify this tissue as a stratified tissue, but as I look at the tissue, I will see that the apical cells are columnar, and I can't have columnar apical cells. I can't have tall cells as the apex, because simply if I was full of more than one cell layer, as I keep going away from the basement membrane, what's going to happen? The cells will become much smaller in size. So looking at this tissue, I have columnar cells at the apex. This will tell me that all the cells are actually attached to the basement membrane. If those apical cells were not attached to the basement membrane, they should be much smaller in size compared to the basal layer. Does this make sense? Does this make sense here? All right, great. Any problem with this concept? What we're saying again is that if you see stratified epithelium, you see multiple cell layers. Look at the shape of apical cells. What are the top cells? Those cells are columnar, they are elongated. I can't have elongated cells at the apex because having those large cells would require me to supply lots of nutrients and oxygen for them to be that large. So being located away from the basement membrane doesn't allow those cells to get nutrients. So this means that these cells are actually attached to the basement membrane, but I can't see the attachment. All right. So what am I looking at in here? What am I looking at in here? It's one row of cells that give me the impressions that I'm looking at more than one cell layer. So this means I'm looking at a fake stratified. Fake stratified, or what we call pseudo. Pseudo means it's fake. Pseudo stratified epithelium. So we will not call it simple, we will not call it stratified, we will call it pseudo stratified. And what is the shape of the apical cells again? Those cells are columnar. So what is the type of epithelium that we're looking at again? It's a pseudostratified columnar epithelium. Pseudostratified columnar epithelium is the type of epithelium that you can see on here. Looking here at this tissue, what do you think? Do I have one cell layer, more than one cell layer? First, how would I know that this is epithelium? What you should be looking for, space. Do I have a space and this space is lined with cells? What do you think? Do you have a space lined with cells on here? Space lined with cells, yes or no? Yes, space lined with cells. So this tells me I am an epithelium. This is what I know. And how would I classify the epithelium? Again, according first to the number of cell layers. Number of cell layers. I have one or more than one cell layer forming this epithelium. What do you think? You see only one row of cells or many cell layers on top of one another? Many. So this is going to be stratified. And what is the next thing should I be using to classify the epithelium? Again, it's going to be the shape of which cells. Which cells should I be considering? Those ones, those ones, those ones. 1, 2, or 3. Apical cells. And when I look at the apical cells, Look closer at apical cells, the cells at the top. What do you think those cells will be? Are those flat cells, cube-shaped cells, or tall cells? Those are flat cells. So this is gonna be a squamous epithelium. Squamous epithelium. So what am I looking at in here? It's the stratified squamous epithelium. Stratified squamous epithelium. Stratified squamous epithelium. Anybody has a problem with identifying here stratified squamous epithelium? First, I need to identify that this is epithelium. How would I know this? Space lined with cells. Look at the cell layers. Do you have one cell layer or more than one cell layer? I would say that I have many cell layers. So this is going to be stratified. Look at the shape of apical cells. What is the shape of those apical cells? The cells at the top layer. Those are flattened. So this is going to be a stratified squamous epithelium. Stratified squamous epithelium. All right, so looking here at my next tissue. What do you think is this tissue? So first thing, do I have the space lined with cells? This is my first question in order to say, in order, so I did mute everybody. Is this all right? I have my neighbors cutting the grass. That's why you might have an ambient noise in the background. I'm sorry for this. All right, so again, what we're looking at in here, we're looking at space lined with cells. Space lined with cells. So this tells me I am looking at epithelium. So what is the next thing I need to look at in any kind of epithelium? I need to look at how many cell layers. Do I have one cell layer or more than one? What do you think? Is this one cell layer or more than one cell layer forming this epithelium? More than one cell layer. So this is gonna be classified as stratified epithelium. Stratified epithelium. Now, what should I be looking at? Look at the outermost cell layer. Look at the outermost cell layer. So what do you think is the shape of the apical cells in this case? What is the shape of this cell? What do you think? Is this cell has a width much greater than the height? Yes, so this means I'm looking at squamous cell. Let's check another one here. What do you think is the shape of this cell? So has a height that is almost equal to the width of the cell. So this means I am a cube. So what I see in here, I see that the apical cells, some of them are squamous and some of them are cuboidal. So Which one should I consider while classifying the epithelium? I will consider both. So this here is a special type of tissue that lines the urinary tract, lines the urinary bladder, ureters, and urethra. So what is special about this tissue is that its cells have the ability to be stretched. So if you have a balloon like this, and you pull the balloon from both sides, how will it look like? It will become stretched like this. So before, the balloon did have a width almost equal to the height. But when I stretch it, what's going to happen? The width will become much greater than the height when I stretch it. So this is a type of epithelium that allows stretch to take place. So for example, this is the urinary bladder. I kept drinking water all day long. So what's going to happen? I'm accumulating urine in the urinary bladder. So this will cause stretch of the urinary bladder. So in order to accommodate this stretch, what should I do? The cells should be flexible. So the cells here are going to be stretched as well. So before the stretch, the cell did look as if they are cuboidal. But when you stretch the cells will become flattened. So what would we call this type of epithelium? We'll give it a special name here. It's a transitional epithelium. What I mean by transitional epithelium, it's an epithelium that has the ability to change the shape of its cells. All right, so why do I have squamous cells and cuboidal cells? Simply because this is a special type of epithelium that has cuboidal cells, but when I stretch them, they will become flattened. So what do we call this type of epithelium? That can change the shape of its cells. It's my transitional epithelium. Transitional epithelium. Is this point clear? Anybody is confused about the transition epithelium? Anybody didn't get how come do we have two different types of cell shape in the outermost layer of the tissue? Any trouble with this concept? All right, great. So let's look at the details here for those different types of tissues that we've covered. So we've seen here space lined with cells, space lined with cells. This tells me I am looking at epithelium. I look at one side. Is this side is lined with one layer of cells or more than one cell layer? I have one layer of cells. So this is simple. What is the shape of the cells? I see those cells are flattened. So this is going to be a simple squamous epithelium. In what scenario would I have a barrier formed of one row of flat cells? if I want to have a large barrier or a minimal barrier? What do you think? A strong barrier or just a very delicate barrier that doesn't create much resistance? If I have just one row of flat cells forming this barrier, you think the barrier I'm creating is strong or not that strong? Compared to if I'm telling you I will build a barrier formed of something like this. Which one is a stronger barrier? What do you think? A barrier formed of one row of flat cells or many cell layers? What do you think? Many cell layers. So here, what should come to my mind when I see simple squamous epithelium? What should come to my mind? That I'm looking at sites where I will need a minimal resistance for me to move stuff across the barrier. You want to move stuff across the barrier, so this barrier shouldn't be that thick. You want to have one row of flat cells to minimize resistance. I have a minimal resistance. So where do you think would you need to have simple squamous epithelium? Where would you need to have a minimal barrier? First location is going to be in the air sacs of the lungs. If I'm looking here, this is going to be the lung like this. It gets air through bronchi and bronchioles that will end up with the terminal alveolar ducts. So, and those are the air sacs. This is one of millions on here in the lungs. So this will be lined with one row of flat cells. Why do you think you're going to have simple squamous epithelium lining the air sacs of the lungs? Because I need a minimal resistance against the gas exchange. So I'm getting oxygen from the air sacs into my blood. I am moving carbon dioxide from the blood to the air sacs. So I need to allow a minimal resistance to allow maximal exchange to take place. What else? Would you need one row of flat cells? Where would you need a minimal resistance? If we're looking here at the inside of the heart, and the blood vessels. If this is a blood vessel, which one do you think will allow less resistance against the blood flow? Just one row of cells which are flattened like this, or cuboidal or columnar? What do you think? Which one will allow the blood to travel with the minimal resistance? Just one row of flat cells. So what's another location for simple squamous epithelium? It's going to be lining the blood vessels, which makes perfect sense because I don't want to have friction all the time. I want to have the minimal friction, the minimal friction possible while the blood is traveling through the blood vessels. So all the blood vessels are going to be lined with simple squamous epithelium. The same exact concept would be applied to the inside of the chambers of the heart. The chambers of the heart are allowing the blood to flow in, flow out. So you don't want to have a lot of friction with the wall of the heart. All right, so what kind of epithelium you think we're gonna have that will allow the minimal resistance against the blood flow again? It's gonna be my simple squamous epithelium. Simple squamous epithelium. Not just squamous, simple squamous epithelium. One row of flat cells. What else would you need to have simple squamous epithelium? If we're looking here at the kidney, what is the function of the kidney? If you know, the function of the kidney will be to allow the blood to get in, carrying wastes, and then will be drained out without those wastes. Where are those wastes gonna be accumulated? They are gonna be accumulated in the form of urine down in the urinary bladder. Until when? Until it gets filled with urine, so a person will feel the urge to micturate, to go to the restroom. So this is basically how the kidney functions. But how will it be able to complete this task? So if we're looking on here, we will see that inside the kidney, we've got functional units. And those functional units are called the nephrons. And the nephrons are here, as you see on here, are gonna be formed of tubules. And those tubules will be acting as a pathway for this filtrate that will become urine later on to travel. So the very first step in order for me to get this filtrate formed is to push lots of fluid from the blood into those tubules through a tuft of vessels, and we call those vessels are the glomerular capillaries, or a single noun, it's going to be the glomerulus. What is the glomerulus or the glomeruli? Those are the blood vessels located inside the most proximal part of the nephron, and the nephron is the functional unit of the kidney. All right, so if we look around here, this is a blood vessel called the glomerulus, and this is the kidney tube that wanna get fluid from the blood. So what do you think? Would you like to have as a type of epithelium lining those blood vessels in order for you to be able to filter this blood and release the fluid into the kidney tubules? What do you think? Would you like to have high resistance or a minimal resistance facing you in order to filter the blood? What do you think? Need to have minimal resistance. So this is gonna be what kind of what kind of epithelium will give you the minimal resistance that you want? Simple squamous epithelium. All right. So where can you find simple squamous epithelium? You will be able to find simple squamous epithelium in the kidney glomeruli. What are the kidney glomeruli? Again, this is a tuft of blood vessels that will be responsible to allow the filtration of the blood. And being in need to filter the blood, I want to have the minimal resistance against this filtration to allow this filtration to take place. Actually, if a person failed to filter the blood because of accumulated connective tissue here or thicker filtration membrane, this can cause renal failure. So it's so critical to have just one row of flat cells to allow this filtration to take place, because any thicker filtration membrane will slow down the filtration, and in some cases it will be completely shut down. Where else? In the air sacs of the lungs. Again, why in the air sacs? Because this is where we are conducting the gas exchange. So in order for the oxygen to travel very fast from the air sacs to the blood vessels, and for the carbon dioxide to travel from the blood to the air sacs, I need a minimal resistance in between. So the lining of the air sacs is going to be formed of one row of flat cells, providing me with a minimal resistance, minimal possible resistance. What else? You want to have a minimal resistance against the blood movement. So the blood vessels and the cardiac chambers are also going to be aligned with simple squamous epithelium. Also, in the body cavities, which we're going to have surrounding the organs. So I have membranes around the organs. Those are called the serous membranes. And let's say on here, if I'm looking at the heart, it has a serous membrane that surrounds it called the pericardium. It has two layers like this. What do you want to have as a type of epithelium in those membranes that are surrounding the heart? To have as a minimal resistance. So every time you move the heart, there will be some kind of friction between those two layers. So what should I keep as a type of epithelium forming those layers? I should have simple squamous epithelium to minimize this friction. So in the serosa, in the serosy here of the body, also we're gonna have simple squamous epithelium. All right, moving on to our next type of epithelium. As you remember, we've seen a space, and this space was lined with cells. So this tells me I'm looking at epithelium. So we did look at just one part. We will find out that I have only one side at a time, that you are formed of one row of cells. So this is a simple epithelium. Looking at one cell after that, we will figure out that it's formed of one row of flat cells, one row of cuboidal cells. So the width of the cell is almost equal to the height of the cell. So this is going to be simple cuboidal epithelium. Simple cuboidal epithelium, it's not concerned with providing a minimal resistance against the movement of substances. It's mainly concerned with allowing secretion and absorption to take place. So what am I looking at in here? Again, this is my nephron. What happens in the glomerulus, if you remember? We did filter the blood. So now we've got fluid out. And this fluid contains a lot of nutrients, like glucose, for example, like amino acids. Would you like to release this glucose out in the urine? So you did eat a donut to get this glucose to get energy. Would you like this glucose that got filtered in the kidney tubules to be released out in the urine, you to get rid of it? What do you think? Hello? Recapture it, right? Reabsorb it. So looking on here, I want to get this glucose back to my blood. How about you've got a toxin or a drug that wasn't filtered in the glomerulus? Would you like to keep it forever in your blood, in your system? What do you think? No, you need to secrete it. So who will be reabsorbing and secreting stuff from the filtrate? This is gonna be the simple cuboidal epithelium lining my kidney tubules. This will allow you to modify the filtrate into urine. So what we're looking here at is simply a cut section like this of the kidney tubules. See this slide? This slide is showing you the kidney tubules, tubes next to one another. Actually, this is what we're looking at. We're looking at a cut section of all those kidney tubules. So what's going to appear on the slide are circles next to one another, like this. This is what we're looking at on this slide. Looking at our next type of epithelium, remember, we've seen one layer of tall cells. This is going to be my simple columnar epithelium. We have two varieties of simple columnar epithelium. Either the apical cells had hair-like projections coming out. We call this as ciliated. Or the columnar cells at the apex don't have those hair-like projections. We call them non-ciliated. So again, what are the two varieties of the simple columnar epithelium? It's either simple columnar ciliated or simple columnar non-ciliated. What is the function of the cilia? Cilia are gonna have the ability to move. They are motile. So where do you think you're gonna need to have ciliated epithelium in your body? First location is gonna be in the airways, small airways. So if, for example, I'm breathing. So here, those are hair-like projections. Hair-like projections. Those are microscopic structures. You see on here, this is under microscope. Those cilia, right? They are not hair. All right. You see the difference? You see the difference? This is not hair, the actual hairs that we have on the skin. This is the one cilia. All right. All right. So this is the type of. This is the hairs that we have in the nose. All right. But here we're looking at hair-like structures. This is very, very, very small. It's microscopic. All right. So we're looking here at where do you think we're gonna need to have ciliated? Here they will allow me to push stuff out. So first location is gonna be the airways, the bronchioles. Right. Those bronchioles are bronchi. Why would you need them to be aligned with simple columnar ciliated epithelium, cells with cilia? If you are breathing air, you're gonna have a lot of dust particles and other particles in the air that you're breathing. In order for you to clear this air, what you're gonna do, you're gonna have an internal lining of the airways that has the ability to produce mucus. And this mucus is a sticky surface to which the particles will stick. But what you're gonna do with the dust particles and mucus that will be accumulated inside your airways, what you're gonna do with them? Would you like to keep them inside the airways accumulating forever? No. What you're gonna do for this mucus, you're gonna push this mucus out by the effect of the cilia. So the cilia will push the mucus, push the mucus, push the mucus. So they will create a flow of mucus that keeps traveling up. So what you're gonna do when you get it in your throat, in your pharynx, you're gonna swallow this mucus with the dust particles to the inside of the gastrointestinal tract. So you're gonna swallow most of this mucus. This is constantly formed and constantly pushed up, carrying all those dust particles. So what happens if a person is smoking? The cigarette smoke will be inhibiting the motility of the cilia. And if I have an inhibition of the motility of the cilia, what's going to happen? I will be, in this case, accumulating mucus inside of my airways. And if this mucus is not moving, this creates a favorable medium for the bacteria to grow. So a person who smokes is more likely to develop infections, lung infections. Why? Because the smoke inhibits the motility of the cilia. So the mucus kept accumulating inside his airways, creating a favorable medium for the bacteria to grow. So, and if the bacteria starts to grow on here, it will cause an inflammation, it causes an infection. So how would I get rid of this mucus that keeps accumulating? And would it be accumulating forever? No, it will be accumulating until it blocks a certain airway. And as it blocks a certain airway, this initiates a reflex, causing a person to cough. And when I cough, what happens? I create a positive pressure within my chest, pushing this mucus to the outside of the airway. All right, so what I'm trying to do here is to create a positive pressure that pushes this mucus, plugging my airways to the outside. Why you don't normally cough, and you shouldn't normally be coughing? Simply because you have simple columnar epithelium, which is ciliated, lining your small bronchi. So why would a person who has been smoking, when he stops smoking, he would be coughing more than when he was smoking? So when he stopped smoking, he will tell you, I stopped smoking, but actually I got more cough now than when I was smoking. So what happens is that when a person stops smoking, the cells will be regenerated, those ciliated cells. And when the ciliated cells are, are regenerated, the cilia are trying now to push this thick mucus that has been accumulated over years. So as I'm trying to push this thick mucus, the cells will get irritated. And as they get irritated, they will start a reflex cough because now I've regained the motility, but the mucus now is too thick for the cilia to move. Where else do you have simple columnar epithelium, which is ciliated? If we're looking on here, this is the uterus. And this is the fallopian tube or the uterine tube. And this here is the ovary. So what happens each month after puberty? Each month after puberty, an egg gonna be released from the ovary into the fallopian tube. The fallopian tube gonna pick up this ovulated egg. And this ovulated egg is gonna be kept in the fallopian tube on here, waiting for a sperm. So normally, where's the sperm gonna be meeting the egg? It's not in the uterus, it's not in here. It's in the fallopian tube. So the sperm meets the egg, allowing fertilization to take place in an enlarged part of the tube called the ampulla of the uterine tube. After fertilization takes place, so now I have a fertilized egg that will be rapidly proliferating into a cluster of cells that will become a baby, right? So this is my fertilized cells that will be rapidly dividing. I have many cells that will become a baby later on. What's going to happen to this morula, to the zygote? Will it be kept in the fallopian tube, or should it be moving to the endometrial cavity, to the cavity inside the uterus? What do you think? Would you keep this proliferating zygote, this zygote that is getting bigger, in the tube, or would you move it towards the uterine cavity? I want to move it towards my uterine cavity, towards the endometrial cavity. So who will allow you to push this fertilized egg? This fertilized egg, who will allow you to move it towards the uterine cavity? It's the simple columnar ciliated epithelium lining the tube and parts of the uterus. This will be guiding the fertilized egg towards the endometrial cavity. What if a female did have an infection? She got accumulated pus, like a gonorrhea infection, for example, which is a bacterial STD, a bacterial sexually transmitted disease. So what's a complication here? It might cause infertility. So no sperm will ever reach because of the accumulation of fibrous connective tissue in here. So I have a total block of the tube. This might cause infertility. What else? It also might allow the sperm to reach the egg, but will be preventing the movement here of the fertilized egg to get implanted in the endometrial cavity. So what this will result in? Baby will start to grow in the tube. Exactly. Tubal pregnancy is a type of ectopic pregnancy. Is this something good to have an ectopic pregnancy, baby growing in the tube? No. Why? Because the tube, it's not that thick. It cannot accommodate that large size of the growing baby. So what's going to happen? Burst. It ruptures. And if it's ruptured, this will cause an internal bleeding. And the only way for me to stop the bleeding is to ligate those large uterine arteries. Means I should be blocking the blood supply to everything for me to stop bleeding. So I will block the blood supply to everything. And if I block the blood supply of an organ, I need to remove it. So the only way to stop the bleeding in this case is going to be by performing a hysterectomy. Why would I consider hysterectomy? Because simply the uterus will not be getting blood supply anymore, because I did cut the blood supply going to the uterus to prevent further bleeding from the lacerated tube, the tube that has burst. So again, back to our epithelium. So where do you think you're gonna have simple columnar ciliated epithelium. We did see two locations on here, small airways and the uterine tubes and parts of the uterus. How about the other type of the simple columnar? The ciliated, the non-ciliated this time. The non-ciliated simple columnar epithelium, it's gonna mainly focus on the production of mucus as a secretion. Also, it's gonna allow the absorption. So simple columnar epithelium, its main function here is to allow secretion and absorption. And where would I need to secrete and absorb large quantities? In my gastrointestinal tract. So gastrointestinal tract all the way from the stomach, so I got the stomach in here, small intestine, large intestines with all the different parts of it, colon, sigmoid colon, rectum, until I reach the anal canal. So all this will be allowing some degree of absorption. Most of the absorption takes place in the small intestine, yes, but all those different parts will allow some degree of absorption, but definitely all of them gonna be responsible to secrete mucus. So for example, in your stomach, you're gonna be secreting lots of mucus that contain hydrochloric acids, that contain pepsinogen, which is a digestive enzyme for the proteins that you release in the stomach. We have a lot of enzymes that got released in the small intestine. Got a lot of mucus secreted in the large intestine as well. So you see on here, where can you find simple columnar non-ciliated epithelium? It's gonna be in the digestive tract, but make sure to read this here. It's from the stomach to the anal canal, because we're gonna see other parts that won't have simple columnar epithelium, but they are part of the gastrointestinal tract. All right, moving on to our next type of epithelium here. As we've seen before, how did we identify this type of epithelium? I look at the epithelium under the microscope, I will see that I am formed of more than one cell layer. So I'm about to say what? I'm about to say stratified. While I go and check the shape of apical cells, what will I find? I find out that those cells are not flattened. They are not cuboidal, they are columnar. So I rethink, can I have tall cells, columnar cells, away from the basement membrane? I can't, because if I am located away from the basement membrane, this means I'm not getting enough nutrients and oxygen. So I can't be that large with getting very limited amount of nutrients and oxygen. So how this works? So we figured out that all the cells are actually attached to the basement membrane. but because they are closely packed against one another, this gives me a false impression that I'm looking at a stratified epithelium. But actually, it is not a stratified, it's a simple epithelium that looks stratified. So some people say we're gonna call it stratified. Some people will say we're gonna call it because it appears as if it was stratified. Some people will say no, it's not stratified, it's simple as all the cells are attached to the basal membrane. So we did agree that we won't call it neither simple or stratified. We will call it, it's a fake stratified, a false stratified, pseudo stratified. And pseudo stratified epithelium in here, it's gonna be of what cell shape? If we are looking on here, pseudo stratified. Looking at the shape of the apical cells, we will all agree that it's columnar. So we're gonna call this epithelium, it's a pseudo stratified columnar epithelium. But this is missing something. I have two varieties of the pseudostratified columnar epithelium, which is actually a simple columnar epithelium, but the cells are closely packed. This is almost the same, like this one. But the cells are closely packed against one another. So I have two varieties, the same way like the simple columnar epithelium. I have either ciliated, so the cells have those hair-like projections, motile projections coming out, or I am non-ciliated. It's very rare to see non-ciliated. You won't see a non-ciliated type on the test, so it's very unlikely. When you complete the visible body courseware assignment, I did find just one diagram that or just one slide that shows the non-ciliated pseudostratified columnar epithelium. All right, so I put it as an example in your assignment, but it's not very common to see non-ciliated. All right, why? It's only limited to the male sperm carrying duct. So we can't see it all over the body. It's just located in the male carrying duct, in the male sperm carrying duct. I'm sorry. All right. How about the ciliated type? Ciliated type is the one that will be lining the trachea. If you remember, this was my trachea like this. It splits into bronchi, bronchioles, small airways that will end up with the air sacs. If you can tell me what are the different types of epithelium in the various locations. So I will mark down 3 parts. All right, so this is part of your in-class activity for today. So the first is going to be the air sacs. What kind of epithelium do I have in the air sacs? This is number 1. Number 2 is going to be the small airways, the small bronchi. This is going to be number 2. What kind of epithelium do I have lining those small airways? And the third one is going to be the trachea. Here, this is number 3. What's going to be the lining of the trachea? All right, so again, this is part of the in-class activity. So please mention the type of epithelial lining in those three different parts of the respiratory system: air sacs, small airways, and trach. So let me answer this with you. What do you think? Air sacs. First one, air sacs. What do you think is going to be the epithelial lining? Simple squamous epithelium, exactly. Simple squamous epithelium to allow gas exchange, to allow free movement of the gas particles. How about, you can on here, small airways. What do you think? Small airways. A type of epithelium that we've mentioned that it will allow you to keep pushing the mucus out. Small airways, small airways. Small airways. Yes, exactly. Simple columnar ciliated epithelium. It's a simple columnar ciliated epithelium. All right, how about the trachea? Trachea, the ones that we've just mentioned. Trachea, what do you think? Is the type of epithelial lining of the trachea? It's pseudo stratified columnar ciliated epithelium. Pseudostratified columnar ciliated epithelium. Exactly. All right, so what's going to be the function of the pseudostratified columnar epithelium? The same exact function, like the simple columnar, allowing secretion mainly of mucus and allowing the absorption. If you notice on here, I have a special type of cells that appears on this slide, and this is going to be, we're looking closer here, a large vacuolated cell. You see, the cell is filled with vacuolated material. It's my mucus cell. Or another name for the mucus cell, here, this is the mucus secreting cell. Mucus secreting cell is what we call goblet cell. So what I can see, so if you got this slide on the test, what is the best identification for the type of tissue? What would I mention? I would write down pseudo stratified columnar ciliated epithelium with goblet cells. All right, this is going to be the best identification of this slide. Pseudostratified columnar ciliated epithelium with goblet cells. All right. Any problem? Any problem so far? All right. Great, great, great. All right. So we're looking here. We've seen space lined with cells. Space lined with cells means I'm looking at epithelium. When I look closer at the cells, I will see that the cells are not arranged in just one layer. I have many cell layers forming my epithelium. Many cell layers forming the epithelium. So this tells me I am a stratified epithelium. Stratified epithelium. Looking at the outermost layer, the apical layer, the top cell layer, I figured out that this is formed of flat cells, squamous cells. So what do we call this type of tissue on here? It's my simple, oh, all right, I'm sorry. It's my stratified squamous epithelium. But you need to remember that you do have two varieties of the stratified squamous epithelium. We have two types of stratified squamous epithelium. You have keratinized or non-keratinized. Keratinized or non-keratinized. What I mean by keratinized and non-keratinized. Let me pull up a picture on. What do you see as a difference between this one and this one? I see a space. Yes, on here, on this slide, you see a space, empty space. Yes. Do I have cells lining the space? So this is epithelium. Same concept on here. Space lined with cells. So this is epithelium. So how many cell layers? Look at the epithelium. Do I have one cell layer or more than one cell layer? Here. Here I have more than one cell layer, many cell layers. So this is stratified. How about this one? See, many cell layers, many cell layers, many cell layers. So this can also be stratified. How about the shape of the apical cells? Where is the apex? Where is the top cells? If I put here 1, 2, 3, 1, 2, and 3, 1, 2, and 3. Which one is the top cell layer? What do you think? 2. Anyone wanna guess? 3. So again, what I'm asking here, where is the top cell layer? Is it 1, 2, or is it 3? Top cell layer, is it 1, 2, or 3? All right, so if I was asking where is the top, it should be 1. But here I'm not asking about where is the top. I'm asking about where is the top cell layer. Cells means they are still living cells. So those are the living cells. They have boundaries and they have nuclei. Those are still living cells. So number 2 actually is the one that is going to be considered the top cell layer. Here, this is the top. Yes, this is definitely the top of the tissue, but this is not formed of cells. What can I see on here? I don't see boundaries of cells. I don't see nuclei. So those are not living cells. This is protein fibers, threads of protein. Those are not cells. They are located on top of the cells. This is my keratin layer. So this one on here, it has keratin layer on top of the apical cells. So what I see on here, I see many cell layers. And what is the shape of the apical cells? They are flat. So this is stratified squamous. This is stratified squamous epithelium. Both of them are stratified squamous epithelium. But here, do I have any keratin layer? Do I have keratin on top of my apical cells? No. But here, do I have those fibers on top of my cells, apical cells? Yes. So what is the difference between this one and this one? This one is a stratified squamous epithelium that is keratinized, or what we call keratinized, stratified, many cell layers, squamous, flat epithelium. All right, so this is keratinized, stratified squamous epithelium. How about this one? What would we identify this one as? This is non-keratinized, stratified squamous epithelium. So again, how many types of stratified squamous epitheliums that we've got? We've got 2 types, either keratinized or non-keratinized. And where did you get this keratin layer? Do you remember? Where, do I have blood vessels in my epithelium? Do I have blood vessels in my epithelium? What do you think? Do I have blood vessels in my epithelium? No, I am avascular. So where do you get the nutrients and oxygen from? From the connective tissue located underneath it. So I get my nutrients and oxygen by diffusion here from the connective tissue. But I am avascular. So as I keep moving away from the base of the membrane, what happened to the cells? See, the cells were columnar. They will become cuboidal. Then as you keep moving up, the cells are shrinking. They will become very thin, smaller, they are flat. So as I keep moving away from the base, cells will become flat. So in this type of epithelium, the cells are going to be regenerating much faster or rapidly dividing. So what happens when the cells are going to be more rapidly dividing? If they are more rapidly dividing, what's going to happen? More cells are going to be pushed up, traveling away from the base. And if they keep traveling away from the base, what's going to happen? They will die. So the keratin layer here, actually, those are keratinized cytoplasmic membranes of dead cells. They kept shrinking on their way up, moving away from the base. And as I am rapidly proliferating, those cells will die and will become keratinized. So what do you think? It's the main function of this type of tissue. Remember, when we spoke about the simple squamous epithelium, one row of flat cells, we did mention that the main function is to provide me with the minimal resistance. Here, having that huge number of cell layers, what do you think? What's gonna be my function? Protection. Protection. So it's providing protection to all the underlying tissues. All right, so where do you think those are going to be located? The keratinized and the non-keratinized. When do you think they will be located? Both are going to be providing protection, but who's going to provide the maximal protection? Who's going to provide the maximal protection? The one that has extra layer of keratin on top, or the one without keratin on top? The keratinized. So where do you need in your body the maximal protection? The part of your body that is exposed to the outside, it gets exposed to all irritation, all traumas, is your skin. So the outermost layer of my skin is going to be formed by keratinized, stratified squamous epithelium. Keratinized, stratified squamous epithelium. Outside of the body. This is where I have the maximal protection. Outermost layer of the skin called the epidermis. So where can I find the keratinized stratified squamous epithelium? It's gonna be in the epidermis of the skin. What is the epidermis of the skin? This is the outermost layer of your skin. All right, where else do I need protection? In anything that gets exposed to the outside, but not all the time exposed to the outside. Sometimes it gets exposed, like what? Like your oral cavity. So if you drink something hot, it gets in contact first with your oral cavity. Your pharynx, your esophagus. So oral cavity, pharynx, and esophagus, those are gonna be lined with a high level of protection, but not the maximal protection. High protection, but not the maximal protection. So they are lined with non-keratinized stratified squamous epithelium. What else gets exposed to the outside, but not all the time? Also is going to be the vagina. So again, non-keratinized stratified squamous epithelium is going to be the lining my esophagus, oral cavity, and the vagina. Eye is going to have skin on the outside. The cornea itself will need to be lubricated, so it's not going to be it's going to be many cell layers. Yes, so let me pull it up on here to show you a picture of it. It's formed of stratified squamous epithelium, but it's not formed of that much number of cell layers. So yes, you're gonna have stratified squamous epithelium, but the stratified squamous epithelium in the cornea of the eye is actually gonna be formed of much less cell layers compared to something like the oral cavity or esophagus. All right, but it's still gonna be considered stratified squamous non-keratinized. Still gonna be considered stratified squamous non-keratinized epithelium. Our last type of epithelium is the transitional epithelium. Can you remind me, how did we identify this type of epithelium? I looked at the tissue. I figured out that we got space lined with cells, so it's epithelium. I look at the cell layers. If I have one cell layer, it's simple. If I have many cell layers, it's going to be stratified. So I see that I have many cell layers, so it's going to be stratified epithelium. So looking at the apical cells, remember, in order to identify the type of epithelium, you need to look at the apical cells. We figured out that some of the apical cells are going to be squamous and others are going to be cuboidal. So which one should I consider? Should I consider squamous? Should I consider cuboidal? Actually, this is a special type of epithelium, like what we mentioned, that allows stretch. And as it allows the stretch to take place, it changes the shape of its apical cells. So we decided to not call it simple stratified or to use squamous, cuboidal, or columnar to identify it. We decided to call it according to its major characteristics, that it has the ability to change the shape of its cells. So we called it transitional epithelium. So again, where is the transitional epithelium going to be located? It's going to be located in the ureters, urinary bladder, and urethra. Urinary tract. All the urinary tract is lined with transitional epithelium. And again, the main function here is to accommodate stretch. All right. Any questions? Any questions? All right, so please answer this as your in-class activity question number 4 for today. Which of the following is not an epithelial tissue? So which one is not an epithelium? We've got simple squamous, yes. We've got transitional, yes. We've got pseudostratified columnar, yes. So the only ones that we haven't seen studying the epithelium, it was reticular. As we're gonna see, reticular is a type of connective tissue. We're gonna see it next class. All right, so please here also in your in-class activity work for today, answer those few questions. All right, so 5, So those other three uh, questions here. So we're going to be stating the type of epithelium, lining, so the first place is going to be urinary bladder. Next is going to be blood vessel. Small intestine.